So in this video, we're going to talk about one of our special senses, hearing and equilibrium, that deal with the ear. So let's begin looking at the anatomy of the ear. And the first thing that I want us to do is make sure we understand that we're looking at the ear based on external, middle, and internal structures. So we have external ear, we have middle ear, and we have internal. Now, the internal ear is going to be fluid filled. The middle ear is air filled, and then of course your external ear is air filled unless you jump in a pool of water and then you can get some water in that part of your ear. We're going to start looking at the external anatomy. So we'll start with the most external, which is the part that you see right on the side of your head, and that's called the auricle. Now the auricle has a function, okay? It's not just to look funny on the side of your head. It actually takes the sound waves, okay, and it sort of funnels them into this tube right here, okay? And that tube it has a name called the external acoustic meatus. So let's look at that. External meaning outside, right? We're getting those sound waves from the outside. Acoustic, that has something to do with sound, we know, right? Acoustic. And meatus means a tube. So this is kind of saying the tube, the external sound tube, right? So those sounds or sound waves are being funneled into that tube. And then what we see is we have this structure right here that you might call your eardrum, okay? But the actual term is called a tympanic membrane. Now I'm going to put a little asterisk right here because this is an important structure in our hearing. When sound waves come in, it's going to cause this to vibrate. And those vibrations, as we'll see, get transferred to the middle ear. It is the division. So we consider that the division between the external ear and the middle ear. Now I'm going to move to a fresh slide so we can look at some of our middle ear structures. But before I do that, I just want to point out that this tympanic membrane then is connected directly to 3 small bones of the middle ear. So in the next slide, we'll talk about these 3 small bones of the middle ear. Okay, now that we have a fresh model, we're going to focus on the middle ear. And there are 3 bones of the middle ear. Remember, this is an air field. These 3 bones are called ossicles. And they are so just remember that it starts with M-I-S. Okay, so they are the malleus, the incus, and the stapes. And so going from the malleus directly contacts the tympanic membrane. So here's our tympanic membrane, right? The malleus is that first small bone that's making contact with the tympanic membrane. Then the incus is next, okay? And then the stapes is the final one. The stapes looks like a stirrup, okay? So this one right here, this last bone that looks like a stirrup, that's the stapes. And then we have the incus in the middle. And then this bone that's directly connected or makes contact with the tympanic membrane is the malleus. Now, how does sound travel in our inner ear, to our inner ear through our middle ear? So as the tympanic membrane begins to vibrate, right, when sound waves come in, that vibration is transferred to that first malleus bone, which begins to vibrate. Those vibrations are transferred to the incus and to the stapes. As that happens, they're actually amplified. So the sound waves are amplified in that middle ear. And then when we look at the inner ear, we'll see how those sound waves get transferred to the fluid-filled part of the inner ear. Now, before we move on to the inner ear, I did want to point out one other structure, and that's this tube right here. Okay. This tube right here is called the pharyngotympanic. Let me just write it out here. The pharyngotympanic tube. So pharyngo for pharynx, tympanic for tympanic membrane. This is important in, like, if you are flying on an airplane and you feel that pressure build up and then you chew gum or you swallow and you get that relief from the air that's moving, that's what this tube does, okay? It's going to help regulate so that you don't have a buildup of too much pressure in the ear portion. So that allows you to equalize pressure. Now, that's a component of your middle ear, right? So remember, your middle ear is air, whereas your inner ear is going to be fluid. Now, one other structure that you cannot see in this, and we'll see it in another image later, but we talked about the tympanic membrane is the division between the external and the middle ear. We also have a division between the middle ear and the inner ear, and that's called the oval window. So the oval window is the division between the middle and internal or inner ear. Okay. Now we'll see the internal ear better in another view, but what I want to point out, we have these little circular things, okay? And these are part of the internal ear. These are called the semicircular canals. And then we have this snail-shaped organ, okay? And this snail-shaped organ is called the cochlea. And as we continue, what we'll learn is that the cochlea is very important in collecting our hearing information, the sound information, and the semicircular canals are going to be very important in our equilibrium and balance. Okay, we have just a different model showing the ear anatomy. And what we can see here, okay, we have the auricle. So see if you can go through, right, and identify. This tube right here is our external acoustic meatus, right? That's where the sound waves are going to be concentrated from the auricle. Now, the tympanic membrane is this gray structure, right? That's our division between external and middle ear. And then we can see these three ossicle bones much better in this image. So we have this first one, I'm going to put an M for malleus, and then an I for incus, and then an S for this little gray stirrup-looking stapes. So those are the three bones of the middle ear, which is air-filled, right? And then we also see this is the tube that's going to allow us to equalize our pressure, okay? If the pressure builds up in the middle ear. This is called the pharyngotympanic tube, right? This also can, if we maybe are sick and we have some mucus buildup in our throat, and some of that bacteria can climb up, begin to divide into this tube, we can get an infection in that tube. So that's not a good thing. Now I'm going to move forward to a fresh, clean image so we can look at the internal structure. So what we can see here, again, we can see the cochlea, right? The snail-looking structure of the inner ear. And we can see a little bit different view of these semicircular canals that exist in the internal ear. Now, we can't see the division very well between the middle ear and the inner ear, but it would be right here where the stapes meets this structure, and there would be something called the oval window. And that's the division between the middle and the internal ear. Okay, in this structure, we're actually looking at close up, right? The cochlea and the semicircular canal. So this entire part is all part of the internal ear. So we want to identify the anatomy of these. So first of all, we have the semicircular canals. So these are all three semicircular canals. Oops, that's a C and a U, okay? And then we actually have these sort of enlarged areas as part of the semicircular canals. And those are these wider spaces, okay? These are called the ampulla of the semicircular canals. And then now we have a very good view right here of the oval window. Remember, this is the division. So that stapes bone, the third of those three small bones, that's where that stapes would make contact. So just as the sound waves, when they meet the tympanic membrane, it vibrates, and then it vibrates each of those three bones. As those three bones vibrate, the vibrations will travel into the fluid through this oval window. And we'll look at a little more details about that in a little bit later. So then we have the cochlea on this particular portion. And if you look at this same structure, right? We're looking at it from another view, the opposite side view. If we flipped it 180 degrees, then one thing I want to point out, it's really nice to see right here. This is our cranial nerve 8. Whoops. Let me try that again. Cranial nerve 8. And this is our vestibulocochlear nerve. And you can see that part of this nerve, as it's traveling back, right, to the brain, part of this is being collected from the semicircular canals and the vestibule. Part of it's being collected from the cochlea. So that's our cranial nerve 8. Now, some other structures I want to point out. This portion here, okay, is called the saccule. And then this portion is called the utricle. Okay, and the utricle and the saccule are going to be very important structures in what we call static equilibrium. So this is related to equilibrium about gravity, okay? And these are, remember, all of the internal ear is going to be fluid filled, okay? So these are essentially like fluid filled chambers that deal with our static equilibrium. So together, these two form, or they make up part of what's called the vestibule, and that's where part of the cranial nerve 8 is named, right, from the vestibule. So one thing I also want to mention about the semicircular canals, they also play a role in our equilibrium. They're involved in what we call dynamic equilibrium, which essentially means your rotational equilibrium. So you notice that they exist in 3 different planes. And as we'll learn in lecture about how this fluid moves and moves through these specialized hair cells, it helps our body to have the information about our position. So those enlarged ampulla, the areas that are on those semicircular canals, those are the places where that rotation or our rotational equilibrium is detected, where that information is collected and sent to our brain. So now we're going to look at internal structures of the cochlea. Now remember, I'm not a good artist, but the cochlea is that snail-shaped looking organ, okay? So if you imagine, right, that this is essentially a tube that's wrapped around itself, okay? And we're looking at a cross-section, right, of that tube anywhere on that cochlea organ. So in this tube, what we find is 3 fluid-filled spaces, okay? So we have a superior, at least on our model, the part that's the superior, okay? The part that's inferior, and then we have this middle or intermediate part. So our superior part is called the scala vestibuli. And then this intermediate part is called the cochlear duct. And this inferior portion is called the scala tympani. Now, those three fluid-filled parts are separated by some membranes that are important for us to know. So the first one is going to be this membrane right here, okay? And this is called the basilar membrane. And then we have another important structure that's called the tectorial membrane. So remember, we already had the tympanic membrane. That's like the eardrum, right? That's between the external and the middle ear. This structure here, this is called the tectorial membrane. So let me just... Now, there are some specialized cells that exist in this cochlear duct portion. And these are hair cells. It's harder to see in this model on the left. The model on the right is enlarged a little bit. So this portion right here, that is the cochlear duct, okay? So the scala vestibuli and the scala tympani, those really, this would be, right, if we continued around, this would be the scala tympani. And then up here, if we had another fluid-filled tube, that would be the scala vestibuli, but it's not really shown on this model. So I need to erase because I have really put a lot of stuff on here. Okay. So we can look at this other model and look specifically at these special hair cells. And in lecture, we'll talk more about all the details about how this works, but we need to look at least identify the models. So this is our cochlear duct, okay? This portion here, oops, this membrane right here, that's our basilar membrane. This in purple is the tectorial membrane. Now, these cells that are shown in green, okay, these are very special hair cells. And these cells are these are the specialized cells that detect sound waves. So at the end, you can see tiny hairs that project from those hair cells. So as sound waves come in, okay, they come and they vibrate that tympanic membrane. And then those three ossicle bones are going to vibrate together. That sound gets amplified. And then that vibration from the air in the middle ear through those three bones is going to get transferred to the fluid. And the first place it gets transferred is to the scala vestibuli. And then it's going to pass through to the cochlear duct, which is this area here. And when it does that, it's going to cause vibrations in the basilar membrane. So this basilar membrane down here is going to vibrate because of those sound waves. Now, that was going to cause these hair cells, right? If what's below those hair cells is bouncing or vibrating, it's going to cause those hair cells to bounce or vibrate. However, this tectorial membrane that's superior to those hair cells, right, that's up on top, it does not move, okay? It does not vibrate with the sound waves. So what that means is, as these hair cells are vibrating, that when they make contact, push up against that tectorial membrane, then it's going to cause those to bend. Those hairs are going to bend. And it's the bending of the hairs then that will eventually lead to opening of ion channels and the sending of a nerve impulse. So that is the part, right, that is important in us actually detecting sound and sending that through a neuron, right? A pathway to the brain so that we can understand and perceive that sound. Okay, so at this point, we've learned some anatomy of the ear. We've looked at some functions and specific structures, but let's look overall, what's going on? How do we hear? How does all this work so that we actually perceive of the sound? Okay, let me get my pen on. So initially, right, we know that sound waves are funneled by the auricle into this external acoustic meatus. The structure number 1, that's our tympanic membrane, right? Those sound waves are going to cause that tympanic membrane to vibrate. That vibration then gets transferred to these 3 small bones, the 3 ossicles. Okay, the first bone that's connected to that tympanic membrane is the malleus, which transfers then those vibrations to the incus, which transfers the vibrations to the stapes, and that's amplified through that transfer. Okay, remember the middle ear that contains those 3 bones is air-filled. Now the stapes then is going to vibrate against this structure right here. This is the division between the middle ear and the internal ear. That's called the oval window. That vibration then transfers from air to fluid, and it begins to move through this fluid, right? It causes the fluid to move. It enters into this first fluid-filled area called the scala vestibuli, and it is going to then enter into the second fluid-filled portion. That's the cochlear duct. Okay, so the one that's shown in purple here, that middle tube, is the cochlear duct. It's in the cochlear duct, right, that the vibrations on this basilar membrane are going to cause the basilar membrane to vibrate, which is going to move those hair cells. But the top membrane, this tectorial membrane, does not vibrate with the sound waves. So those hair cells then, as they bump up against that tectorial membrane, are going to bend, right, as they bump up against it. And it's the bending of those hairs on the hair cells that's eventually going to cause the impulse, right, to be sent. And that's how we perceive of the sound. Okay? Now that's going to travel, right, through our cochlear nerve, the cranial nerve 8, the vestibular cochlear nerve. Now, one thing I haven't mentioned is what happens then. Well, those sound waves eventually enter into this third fluid-filled space. Okay, this is the scala tympani. And then those are going to exit the internal ear through what's called the round window. So essentially, that's when we don't want to continue to hear the same thing echoing over and over and over. So that's the way that those, once they're passed through the cochlear duct, they exit through the scala tympani so that we don't continue to hear the same sound over and over again.